Next up, we have Dr. Christine Lee from the NASA Jet Propulsion Laboratory.
I also have a short person challenge there. Hi, thanks for having me. So you'll see that the title of my talk is a little bit different from what's actually listed in the program. And that's because I wanted to talk about the entire project that covers not just turbidity, but other water quality parameters as well. We were selected for an award about a year and a half ago through NASA to try and improve the utility of different types of remote sensing data on enabling water quality monitoring, water resources management, focusing on California's water systems. And so we have been working rigorously towards that, and I'm happy to share some of those results with you today. And so here is the obligatory NASA Earth Science fleet of all of the different Earth Science satellites that we have currently orbiting the Earth. There's over 25 of them, and of those 25, about 17 of those spaceborne sensors can be used to better understand different types of water cycle variables. These satellites are put into space to help us understand Earth system science and to advance our knowledge of how the water cycle is changing. So looking at meteorology, but also looking at land use and other parameters as well. And so one of the challenges in trying to leverage remote sensing information lies in the fact that these systems are designed to acquire data on a global scale. And so they're designed to be able to cover the Earth at a certain temporal resolution. Depending on what parameter you're focused on, there are different types of resolutions that we're concerned with, such as spatial or spectral sensitivity and so forth. So through the combination of airborne and satellite remote sensing, we can get a better understanding of how water cycle processes are changing on a global scale. But of course, water resources management decisions are not always conducted at that scale. It's conducted at a much more localized regional level. And so many of these decisions rely upon ground-based observations, modeling to help inform decision-making at those scales. And so the question is, how do we combine these data sets and leverage the in situ networks and also leverage the remote sensing to build an enhanced water quality monitoring program? So similar to what I was just saying, a lot of these remote sensing systems are designed for different types of parameters, but we still feel that we can make a substantive contribution to water quality monitoring by leveraging them. And so, for example, a lot of the parameters that we can resolve using satellite data might be designed to study open oceans. And the trade-off there is that we might not get the spatial resolution that's needed to look at smaller inland water bodies, resolve coasts. Another really popular or commonly used platform is the Landsat series, which is considered a land imager. And so some of the trades there are that we might not get the spectral resolution or the sensitivity that we need to really resolve aspects of the water column. But by looking at a suite of these instruments, we could still develop water quality parameters that could be potentially useful for improved monitoring. And so the project that we've set out to do is really one of trying to increase accessibility and also see where remote sensing data could be used to support water quality monitoring, and then to demonstrate those utilities by partnering with different decision makers and agencies and define use cases with them. And so we're working with a variety of partners, several in academia, so UC Merced, Erin Hestir is the co-investigator on that program, and you'll hear from her next. We're also working with Oregon State University, but a couple of our key partners are the Metropolitan Water District and the California Department of Water Resources. And we continue to have additional conversations to try and expand that kind of partnership group. And I also wanted to mention 34 North, who we've been working with as a way of getting our data archived and available more broadly through the Bay Delta Live portal. As I mentioned, we're focused on California's water systems, so the 3 different sites that we're focused on in this project currently are the San Francisco Bay and Delta, Lake Oroville, and San Luis Reservoir, as we've noted up there. Okay, so these are the 3 water quality parameters that we're currently focusing on for this project. They are turbidity, chlorophyll, and water surface temperature. We picked them because they exist at kind of this perfect intersection between being able to be reasonably resolved using satellite and airborne remote sensing, as well as being management relevant. And so some of the different types of management decisions that we're trying to tie these data with include managing for minimizing smelt entrainment, improving delta smelt habitat, looking at export of phytoplankton and the actions that control that, as well as precursors for harmful algal blooms to minimize public health risk. And then lastly, looking at thermal risk of these endemic species. And so just a brief overview of some of the principles behind our methods. And so the way that we measure ocean color or water quality information is by looking at optical properties of the water column near the surface. And these are governed by what we call inherent optical properties. And so these are parameters that you can actually collect if you were to go out on a boat or install instrumentation on a buoy. These are measurements that you can take while out in the field. And the actual amount of absorption and backscatter that occurs in that optical water column is dependent on the parameters that are in there. So for example, looking at the amount of sediments in that water column might change those properties, looking at dissolved organic matter, algal particles, and so forth. And then from the satellite or airborne sensor, we can then derive remote sensing reflectance from the air. And so by relating these two different measures of remote sensing reflectance, we can then begin to develop these algorithms for estimating things like turbidity, chlorophyll, turbidity and chlorophyll. Temperature is a little bit different. And depending on what your water body looks like, you might find that whatever is dominating that water column at a particular area can be resolved by looking at different remote sensing reflectances. And so in an area that might be dominated by chlorophyll, you can actually see that response in the reflectance spectra, as you can see here. And so by looking at different pieces of information across these spectra, we can begin to derive information from those and infer these water quality properties. And so turbidity is the first parameter that I wanted to share some results with over the delta. And so one of the key elements of the project are seeing how well the remote sensing derived measurements of these water quality properties relate to ground-based measurements, because ultimately we want to show how well these algorithms perform relative to what the management-based standard is. And so what we do here is we take the satellite-derived estimate of, for example, turbidity. We extract those pixels those pixels that are over the stations using the station lat longs, and we're getting data from CDEC. So all of the all of the turbidity data that are available through CDEC. And then we also aggregate the turbidity data from stations, and then we do a direct match of a comparison, and we have so far been looking at regressions across the delta. And so I know this might be hard to read, but this table is just to kind of summarize some of the key differences between the two different types of data sets. And so when you're looking at the water quality stations, you're primarily working with point data. You might have on the order of 100 stations that are distributed throughout the San Francisco Bay and Delta, which is actually considered very well instrumented. But it is meant to represent, over 5,600 square miles of that system. But it also has a very high temporal resolution, so taking data every 15 minutes. On the flip side, when you're looking at remote sensing data, for example, with Sentinel-2 or with Landsat, you're getting an acquisition every 4 days or maybe every 2 weeks. And so in comparison to the 15-minute aggregation, you'll see we estimated, or we counted up about 55 clear day incidences between 2016 and 2018. Instead of point data, you're looking at pixels, so an average over, for example, 20 meters by 20 meters. And then instead of 63 to 100 stations, we're now looking at 36 million pixels or points that you could potentially use to better understand the water quality conditions of that particular acquisition. And so this is what it looks like when you dump all of the data over 3 years, comparing the satellite-based turbidity on the y-axis and the CDEC turbidity values on the x-axis. I put the horizontal and vertical bars just to kind of show where the kind of most relevant NTU values are for the delta smelt, so at 12 NTU and at 80 NTU. But I was actually very encouraged by this, even though it might kind of look like a mess to everybody else, because it showed us that there was at least an order of magnitude, there was a reasonable estimate that we could derive from the satellite data in comparison to what we were seeing on the ground. And so we started to divide up and try and understand what would be causing the scatter in that previous plot. And so one of the first things that we did was look at these relationships by station. I know this is also hard to see, but here's a sampling of stations. In the middle is the map of which stations were selected for these different panels. Again, was very encouraged to see that they were actually hovering pretty close to the one-to-one line. But you'll see that there is quite a bit of site-to-site variability over this 3-year period when comparing the satellite results to the CDEC results. And so we can continue to try and understand this. And one of the goals of being able to understand this is so that we can then say, when we're delivering the satellite data to 34 North to be archived on Bay Delta Live, we can actually provide more information of, when the data could be applied for certain uses, what are the kind of constraints around that and limitations around that. As I mentioned, we're also looking at chlorophyll A. And so the example that I showed here is showing the change in chlorophyll extent in 2017 as it matched up with the different public advisories that were put out, that were put out by DWR to warn the public that it was risky to swim or to go out into San Luis Reservoir. And one of the kind of key takeaways from looking at this was that you actually see an onset of an expanded bloom, or the onset of a bloom occurring prior to when the notification started going out. So can we work with DWR to see if some of these satellite data could be used to support some of their advisories and other updates and notifications around this issue? And so we've been talking with DWR to establish just that. And also beginning conversations for Oroville as well. This is just to mention that we have a proposal out to also look at harmful algal blooms in Southern California reservoirs in partnership with MWD and EPA. Still waiting to hear from that, but if funded, this would be really neat because we would be not only enabling earlier detection of harmful algal blooms, but the UCLA component would allow us to actually do on-site point of detection of toxic species that are present using a deployable flow cytometer that also leverages some machine learning to detect those harmful species. So this is the last product that I'll mention. So we're also, of course, looking at water surface temperature. This has a lot of implications for thermal stress on different fish species. Not just in the Delta, but in other water bodies in the State Water Project as well. And the two spaceborne instruments that we're looking at for this are Landsat 8, which is satellite, and EcoStress, which is an instrument on the International Space Station. And we're looking to see, of course, how these water surface temperature measurements line up with the CDEC temperature measurements, and as well as trying to understand whether they can be used for assessing ecosystem restoration outcomes and export control actions. And so here are some of the early results from comparing Landsat 8 to the CDEC and bulk temperature measurements, as well as looking at the 5-year record from Landsat 8 and looking at a time series. And so we want to continue to dig into these data to see if we can now drive better understanding of what's happening within the systems. I saw a paper that was published pretty recently that did a really nice job characterizing the upper thermal tolerance of the delta smelt, silver side, and an invasive bass, and showing that the silver side and bass had a much broader kind of range for thermal tolerance than the delta smelt. And so kind of the next set of figures and analyses that we would like to do is to actually produce maps of those, that thermal safety margin that corresponds with those 3 species as well. So here's just a quick snapshot, one from winter, one from summer, and you can see that in that right panel figure that there are a lot of areas where in the kind of yellow to red, which is approaching the maximum thermal tolerance of the delta smelt. I mentioned EcoStress, which is an instrument on board the International Space Station, which is actually sampling any given point at different times of day. And this will allow us to do a better characterization of what we're measuring as surface temperature versus what's happening in the bulk temperature, which is what CDEC is getting at. Okay, so as I said, this is all underpinned by the fact that we want to try and build a platform that is an enabling platform that can then allow more people who are interested and could be benefited from using remote sensing data to access those data easily. And so I'm happy to take questions if I can, while this is playing, if I can get it to play. And so this is just one example of the dashboard functionalities that 34 North is building using remote sensing data. And so one of the final deliverables of our project is to have all of these data sets archived on Bay Delta Live, but also to establish that mechanism for them to continue automating the production of all of these data sets as well. And so this is a dashboard that matches up the satellite data to the CDEC stations. But if you're familiar with Bay Delta Live, they have an entire kind of suite of different water quality products and other types of water data that are available there. That was probably mine. So thank you, and I'm happy to take any questions.
Thanks, Christine. Can you talk a little bit about the spatial and temporal resolution issue? So, we're looking at, changing species distributions at the statewide level, and what we really need in temperature is sort of the ability to look at the smaller creeks and streams at a daily time step to get, moving averages. And so that's been really one of the limitations, and has driven us to use modeling air surface temperature and translating to water surface because of those spatial and temporal limitations.
Yes, so that is one of those are the trades that are made early on in designing some of these satellites, is that depending on what it is that you want to focus on, we have to trade off sometimes on the spatial side, sometimes on the temporal side. But we do actually have an opportunity to provide input into kind of the next generation of NASA satellites that are in development. And one of them is called the Surface Biology and Geology Designated Observable. It's a NASA speak for Anyway, and so that is actually slated to be one of the next NASA Earth science missions to be formulated, developed, and built and launched in the next, optimistically, 7 years. And it does have a thermal component to it. And so if you'd like to provide that level of feedback on the needs as far as the spatial requirements that are needed, as well as the temporal requirements that are needed, we can actually, it's the perfect time to feed that back into that design process. But we're aware of the constraints.
Thanks, Christine. That was really neat. I'm wondering if some of the scatter that you're seeing is due to scale differences between what you're measuring with the satellite and what you're measuring on the ground. Scale differences between.
Oh,
I mean, are you possibly
yes.
is some of that scatter possibly due to just smaller scale heterogeneity in the measurement that you couldn't possibly avoid with the water samples on the ground?
Yeah, so it's important to stress that these are not the same types of measurements.
Yeah.
One is an average over a pixel, and one is obviously a point measurement. We're also hypothesizing that another issue is the fact that a lot of these stations are really close to land.
Mm-hmm.
And so you get an effect that is due to being close to land, whether it's scatter from the land directly itself, or if we're actually selecting pixels that somehow are mixed pixels that are intersecting with land. And so those are all issues that we need to look into further.
Because all else being equal, you might come to the conclusion that, gosh, the satellite measurement could be a better measurement of the turbidity in the water than scoop sample.
Our messaging is such that we're complementary
Okay.
with the point data. But depending on what your application is, one may be suited more appropriately than the other.
Yeah.
Thank you. All right, well, my info is up there in case you want to follow up. Thank you.
Next up, we have Erin Hestir from UC Merced. Oh. There we go.
Having a short paper comments. Right. Thank you, Dr. Lee, for that presentation. That was excellent. And I would like to point out to the community here, for those of you who are aware of some of the hyperspectral remote sensing that's been going on in the Delta for the last decade, led primarily by UC Davis, the SBG, this Surface Biology and Geology mission that Christine mentioned, will be a hyperspectral type Landsat. So if you're interested in those applications more broadly, Dr. Lee is the applications point of contact for NASA. So please get in touch with her beyond just temperature if that's of interest to you. So we're all very excited for that development. So I stand between yourselves and lunch and myself and lunch, so I will try to be quick. And I will preface this by saying I am not a geochemist and I am not a mercury expert, so I will do my best to answer questions about the mercury side of this. I am a remote sensing expert, so grill away if you'd like on that. So this project that I'm about to describe was recently funded by the California Department of Fish and Wildlife under the Proposition 1 funds. And we have several colleagues who've been thinking a lot about this, including a group of folks from the California Water Science Center and several scientists from the Menlo Park Center as well. And the basic premise here is that we are going to try to help develop optical proxies for mercury in the water column to help enable more efficient and cost-effective monitoring to complement the regional monitoring program in their efforts in developing and TMDL and managing mercury in the Delta. So some of you have seen these, wow, this is like, that's much better, okay. Some of you have seen these warnings out there in the Delta about eating fish in the Delta and advisories against doing so. A lot of fish around sport waters contain mercury. However, in the Delta, people who are sport fishing are disproportionately consuming those fish compared to other places in the state. And indeed, it's estimated that these people are actually taking in more than the daily recommended EPA dose for mercury. And many of these households that are consuming this fish come from disadvantaged communities and disproportionately have women of childbearing age or children in the house who are potentially consuming these high levels of mercury. So the Delta is a place where we want to start to really focus some of our efforts in having well-informed advisories so the public can make good decisions about fish consumption. So methyl mercury in particular is of concern when we're starting to think about developing these advisories and managing for mercury in the system. Methyl mercury has a bioaccumulation factor that is incredibly high, meaning it moves up through the food web. And we've been using largemouth bass as an indicator for methyl mercury in California surface waters. The two scatter plots on the top here show the methyl mercury concentration in fish flesh, and it shows that it correlates relatively well to the aqueous methyl mercury. And so fish tissue is considered one of the main monitoring mechanisms for methyl mercury in the system. And these lower sets of scatter plots come from a paper that shows that juvenile largemouth bass and silverside fish mercury concentrations correlate very well with the bass concentration. So what we're seeing is that these are good indicators of where mercury is entering the food web. There's also been some concern that some of our attempts to restore wetlands around the bay and delta may be increasing mercury transport and potentially serving as production for methyl mercury in these systems. So what we're seeing on the left here is an interpolation of the sediment concentrations of total mercury from Alviso Slough down in the bay. And this study showed that as you restore a wetland and you start to get some of those nice channels that you'd like in a wetland, that scour also may remobilize some of these legacy mercury that occur in the sediments. And if this occurs in a wetland, these wetlands may have some of the biological conditions necessary to methylate that mercury and really amplify it in the food chain. This is unpublished data from my colleague Mark Marvin de Pasquale that shows that we have much higher rates of methyl mercury in wetlands relative to some other systems from the South Bay. However, data on the effects of wetland restoration are limited because measuring mercury is difficult. There's also evidence that spatially the concentrations of mercury around California and around the Delta are not consistent. And there are some observations that there's been some areas of persistent low mercury relative to others in the Delta, but we don't fully understand what these transport mechanisms are. And if we're going to get to a place where we can fully understand what the spatial and temporal patterns of mercury are so that we can create those well-informed advisories for the public as well as start to effectively manage mercury in the system and understand how to restore our wetlands in an effective way, we need to start thinking about developing cost-effective monitoring technologies that's going to help improve our understanding of where mercury is, how it's being produced, and where it's going. So there's been a lot of work done, a lot of that led by the USGS at the California Water Science Center to conduct high-frequency monitoring. And these new high-frequency sensors create both temporally and spatially detailed water quality data, and many of these are underpinned by optics. So what you're seeing here in the middle is one of these buoys that you can deploy that creates very high-frequency 15-minute data, as Dr. Lee alluded to, for a range of non-mercury water quality metrics that we all have been using and talking about with the CDEN and other data sets. You can take these same instruments and you can deploy them on a boat with a flow-through system so that you can get very high spatial resolution transect mapping of surface water quality. You don't get the temporal resolve, but you get that spatial resolve over the course of a cruise. We then can use satellite observations to fill in these gaps that we have from the point high continuous frequency sites and those very detailed spatial transects to help us give that overview, that picture of high spatial information. So what we're looking at here is a map of total suspended solids concentrations estimated in the delta. This is from the summer of 2015, where the French Space Agency deorbited one of their operational satellites into a lower orbit and acquired images of the delta every 5 days at a 10-meter resolution. So this is an interesting experimental data set for us to start to understand what high temporal frequency, high spatial resolution remote sensing can do to start to fill in some of those gaps. So I'm just going to take you through a time series, watching how the total suspended solids concentration progresses and moves up and down the lower estuary through the course of the summer. So Dr. Lee gave a really nice overview of how we get to water quality from space, and so I won't belabor this point too much. But essentially, what we're taking is the remote sensing signal. So these are the apparent optical properties. This is the radiance and reflectance that is emitted from the water surface. And we are then linking those to the inherent optical properties, either through those models, the BB over A plus B that Christine showed, or through machine learning techniques and other statistical approaches. And then we can back out what the physical biogeochemical properties are. So we're estimating backscattering from the remote sensing signal, and then we're using that to get to total suspended solids. So this is a nice image from the Venetian Lagoon that has kind of everything you'd want in one system. So it's a pretty image to show both what the suspended sediments in the system might look like, the dissolved organic substances that cause absorption like organic matter, algal activity which both absorbs and scatters in the water column, as well as shallow waters which can create reflectance due to the benthos. Now, when we get to these physical and biogeochemical properties from the inherent and optical properties, often these transfer coefficients to get there are regionally and temporally dependent. We are not yet at a state where we can do this globally with one consistent algorithm. And so working at this regional level to a level of accuracy that matters for a given end use case is where we are still with the community. And so thinking about those transfer coefficients, here's an image of Mercury, not quite from space, but from an airplane. And so what we have done here, this is work led by Cedric Fichot, who at the time was a postdoc at JPL. And the JPL flew this very high quality sensor called the PRISM sensor, which is a nice experimental sensor for us to begin to develop these algorithms over the San Francisco Bay Delta. And they took this relationship that they developed from in situ measurements between filter passing methyl mercury on the x-axis, on the y-axis, and dissolved organic carbon measured from point measurements and then analyzed subsequently in a laboratory. And they took that model and applied it to the remote sensing imagery using a regionally dependent transfer model to create our first remote sensing image of filter passing methyl mercury. This isn't from space yet, but hopefully one day we'll get there. So if we start thinking about these relationships, in order to get to that Mercury from space, what we really need to do is we really need to start thinking about these relationships between Mercury and other non-Mercury water quality metrics. So what we are seeing here are two scatter plots. The first on the left is many samples, about 355 samples, collected over a variety of different seasons and different years from April 2005 to June 2011. And this is across many different environments in the San Francisco Bay and Delta. So these are lentic environments, wetland environments, open water environments. There's some relationship going on between these constituents, but it is not quite as simple as just a linear model. If we consider only the open water sites in this, we start to see a much more straightforward relationship, although we can certainly talk details about log-log transforms and how we might want to model that more intelligently. So encouragingly, if we take a look at this figure again on the left, what we're looking at is, again, on the y-axis, the filter passing methyl mercury concentration. And on the y-axis, we're looking at absorbance at 254 nanometers. This is an optical proxy for dissolved organic carbon. And very encouragingly, that A means that it's an absorption term, which means that it's more directly linkable in that remote sensing model that we're working with from a physics perspective. And you get a straight up linear relationship with a much higher R squared. So we're starting to think that this might be feasible. Now, we're also interested in the total mercury that's evident in the water column as well. This is the filter passing total mercury on the y-axis on our figure on the right. And we see that there is a linear relationship there too. Furthermore, we are also interested in particulate mercury. And there is some evidence that it might make sense to actually think about measuring mercury from a gravimetric perspective rather than a volumetric perspective, because you're actually saying how much of the particulates are mercury, which might give you some more indication about what's going on in the food web and how that's bioaccumulating. This is data, again, taken from that Alviso Slough site, that restoration site in the San Francisco Bay, where we are seeing the red dots are the upper location in the slough, and the blue dots are the lower location in the slough. And this is made, these measurements are made across many different days over the course of a year, across different seasons. And what we see is a very strong linear relationship between both the particulate total mercury concentration and the particulate methyl mercury concentration in the water column, and the total suspended solids in the water column. And the mercury is highest when you have low total suspended solids. Basically, when conditions are quiescent, we're seeing when we have the most particulate mercury in the system. So these relationships between different mercury species and different water quality concentrations can be predictive, but obviously there is a lot of variation. Each one of those very kind of simplistic regression models that I showed you had different coefficients, it had different performances, and while they were robust across, a large range of conditions, there's a lot of work to go to really start to model what this is going to look like. And so this gets to what we're starting to do in this project. We're going to co-locate 4 continuous high-frequency monitoring sites at 4 regional monitoring program stations in the Delta, where we will begin to make these better quality predictive relationships between the optical remote sensing, as measured from both in situ sensors, hyperspectral drone sensors. We can talk about that another time. And satellite overpasses to, again, improve those remote sensing products that Christine's project is starting to develop the workflows for. And then we can start to also develop those those better predictive models that are more sophisticated than just a regression between both our particulate and our filter passing methyl mercury and total mercury, and the non-mercury water quality measurements that we're making from these high-frequency instruments. So things like total suspended solids, but also optical backscattering, which is measured in the instruments and is much more relatable to the remote sensing. Dissolved organic carbon, measured in the laboratory, but also measured through fluorescence dissolved organic matter sensors, which is, again, or absorbance, which is much more related to that remote sensing mechanism. In addition to those high-frequency continuous monitoring stations, we will go out and we will do full tidal cycle sampling events where we will actually measure over the course of an entire tidal cycle how these relationships vary, and we will be doing these spatial transects with matchups with the satellite data. So we're going to build this very big data set in order to truly capture the range of variation so that we can make a more predictive and robust model that will enable us to have optical proxies for mercury in the Delta. So in order to tackle this challenge, we're going to use optical sensing. This can provide rapid, low-cost monitoring technology that's going to really strengthen the rigorous regional monitoring program. This isn't going to replace it. We call these proxies, and we believe they will be proxies. So these high-quality sampling programs need to continue to really make sure that we're in compliance with TMDLs, but we hope that these technologies will help fill in the gaps and provide a first-order estimate of what surface mercury is doing in the water column. And again, we really hope that by mapping this mercury, we're going to start to tackle some of those science questions around transport and production, and ultimately serve many of the disadvantaged communities that rely on this information to make informed decisions about their fish consumption. With that, I will take any questions, or if you want to take off for lunch, I can just hang out and answer questions while you all eat.
I'm curious, how do your two variables that are explaining the mercury, the total dissolved solids and the total organic carbon, how did those relate to one another? Are they also linearly related in a positive trend, or are they opposite?
There is a whole bunch of covariation between all of these variables, both the suspended and the dissolved matter. And then the one factor that I didn't bring into this that we're also going to be looking at is chlorophyll production, because that determines some of the organic matter concentration and particulate organic matter concentration, but not all of it. And they are probably also important for part of the mercury methyl production. So it is a much more complex linear problem, or a nonlinear multivariate modeling problem than I've painted with some pretty regressions. So yes, and fortunately we have the biogeochemists and the geochemists on our team, mostly at USGS, who are going to help us tackle those issues.
Cool, thanks.
Hi, my name is LaDonna Williams, and it's good to see your project actually focus on disadvantaged communities. In the past, we had been working on fish consumption in our area here. We're in Vallejo, so between Vallejo, San Francisco area, on the advisories. And then we worked with another organization on testing the fish. But what we noticed is the fish that they targeted was not fish that African Americans ate. So I was just wondering, are you set on the type of fish that you guys do test, and are you open to adding additional species or fish to that list?
So excellent question. This is beyond my expertise, so I will give you a half answer. I'm a scientist, and I'm a remote sensing scientist, so I'm not actually in charge of those fish monitoring programs. However, there are several people here who are involved in the regional monitoring program who can probably speak up to that if anybody is here who wants to raise their hand. However, I think the evidence is that basically, based on those first sets of graphs that I showed, that what we see in that indicator species, the bass, is reflected when they test other fish species. And so they feel like it's a pretty good indicator across many species in the system. But certainly from a human health perspective and really targeting food sources, I'm sure quite a lot of work will be done. We're not really focused on the fish. We're trying to develop these sensors that you can just put in the water to directly get an idea of the concentration. But that is certainly an area of concern, because ultimately we're doing this for fish, wildlife, and human health. So, I'm definitely interested in hearing if anybody knows more about that.
Yeah, I can say a few words about that. Hi, LaDonna. I don't know if you remember me from the Fish Mercury project back around 10 or 15 years ago. We worked together on that one. Yeah, I'm the lead for fish monitoring for the Bay RMP and the Delta RMP. And in the Bay RMP, we have the funding to look at a variety of species, and we work closely with OEHA, the Office of Environmental and Health Hazard Assessment, to provide data to add more species to the advisory for the Bay. For the Delta RMP, the budget is a little leaner, and so we're focused on just basically one species, the largemouth bass, because it's a great mercury indicator, and we don't have the funds to look at a variety of species throughout the Delta. I'd like to catch up with you and hear about what you've been up to in the past decade or so, and we can talk more. Yes, Jay Davis. Yeah.
Thank you, Jay.